Good morning, good afternoon, and maybe good evening for some of you. Welcome to the International Plant Breeding Seminar Series. Today, sorry about the mistake on the title there. We have Alexandre Aono, who's going to talk about plant genetic diversity integrated strategy for understanding, exploiting, and conserving it. This is live from last seminar, so disregard the title. But Alexandre is advanced PhD student at Universidad de Campina in the state of Sao Paulo, and University of Campinas, and his research is focused on integration of omics and genomic prediction methodologies for plant breeding purposes. He spent 1 year at the Roslin Institute at the University of Edinburgh there, where he gained a lot of experience in the latest developments of the field of quantitative genetics applied to plant breeding. Alexandre works in the lab of Dr. Annette Pereira, who was originally the person who was going to deliver this seminar, but Annette has some issues, family problems, and she was not able to join us and present. So, Alexandre, I'm gonna thank you so much for accepting the challenge, I would say, and I would give the floor to you.
Let me share my screen, just a moment. Okay, so good morning, everyone. It is with great pleasure that I'm going to present to you a little bit of what I've been developing throughout my graduate years. But before I begin, I would like to acknowledge the organizing committee for such an invitation, and also my supervisor for providing me with the opportunity to share some of our research to you. So, as Carlos presented me, my name is Alexandre Aono. I'm a PhD candidate at the University of Campinas here in Brazil. And I'm going to present to you some strategies that we have been developing to investigate plant genetics in our lab. So my research has been conducted under the guidance of Professor Annette Pereira de Souza, who established her laboratory in the late 90s. So her research has primarily focused on exploring the genetic diversity of several species, with an emphasis on breeding and ecological purposes. Currently, I work with a team of very talented colleagues, which are focused on different plant species, including grapevine, mangrove, sugarcane, rubber tree, and different species of forage grasses. On the other hand, my research lies in the field of bioinformatics. So I obtained my first undergrad degree in science and technology from the Federal University of São Paulo, where I started to develop an interest in the field of bioinformatics. During these years, I had the opportunity to conduct undergrad research in plant genomics and microbial metagenomics. And then in 2017, I started my bachelor in computer science at the same university, and I started to do research using more advanced methodologies for data analysis, including operations research with genetic algorithms, machine learning techniques, complex networks for biological networks, of course, and image processing techniques with deep learning also included. So this enabled me to work with diverse types of biological data sources, which motivated me to pursue a PhD in genetics and molecular biology, with a particular focus on bioinformatics. So in regard to the ongoing focus of the research of our laboratory, diverse questions have been investigated, and of course, depending on the specific plant species under consideration. So despite the shared interest in defining and characterizing associations between genotypes and phenotypes, it is important to acknowledge that each plant species presents its own unique set of challenges. And by starting at the genome level, we can identify and define physically linked polymorphisms. These regions can then be used to estimate the impact of genomic effects on phenotype configurations. However, as you know, the definition of a phenotype requires a more complex set of molecular interactions. So, for example, at the transcriptomics level, transcripts can be identified and used to compare the level of expression of samples under specific experimental conditions, allowing, for example, the investigation of contrasting phenotypes. Furthermore, it's also possible to model the way in which these transcripts interact, defining specific biological functions. And the same observation can be performed for proteins at the proteomics level by contrasting protein expression conditions and examining protein-protein interaction networks. We can investigate specific molecular dynamics that might be related to phenotype differences, including, for example, co-expression, domain relatedness, and also metabolic associations. Finally, the metabolites are considered the intermediate phenotypes, being chemical intermediates linking the genotype and phenotype. Such compounds can then serve as measurable indicators of the biochemical pathways that are activated in response to environmental stimuli. So in the same way, we can contrast their abundance and model their interactions, gaining further insights into the mechanisms underlying complex biological processes. Although investigating these individual omics layers has provided diverse insights into specific phenotypic responses, there is a growing interest in studying the complete set of interactions that shape a phenotype. And this is exactly the focus of my primary research interests: how to integrate diverse molecular layers to investigate the phenotypic configuration of complex quantitative traits. By bringing together diverse molecular data, we can gain a more comprehensive understanding of how these traits arise and how they function at the molecular level. So, for example, if we consider a single SNP, a single nucleotide polymorphism, that is located within a gene, we can then associate such SNP with a transcript, which may be translated into a protein. This protein might represent an enzyme that can facilitate the association of two metabolites. Additionally, this entire mechanism that I described to you can be modulated by bacteria or even influenced by an epigenetic mechanism. And moreover, apart from this series of reactions, the final phenotype can also be shaped by environmental factors. And using such an integrative view, our research group has gained a deeper understanding of diverse aspects of various plants. So, for example, we have investigated how a mangrove species responds to climate change, and also, for example, the molecular mechanisms of grapevine genotypes, how can they impact their phenotypes. So in line with this direction, we have investigated rubber tree growth using GWAS and network-based approach. So regarding rubber tree, these are diploid species with their center of origin in the Amazon rainforest in Brazil, and they serve as a major source of natural rubber. This rubber extract from these trees is a raw material for almost 50,000 products. However, the genetic makeup of such trees is rather complex, with around 70% of their genome consisting of duplicated regions. And such a complexity has posed a challenge in studying and understanding the underlying mechanisms of their growth and development. Additionally, their genetic breeding represents a time-consuming process that has taken up to 30 years for the development of new varieties. In this sense, the use and incorporation of genomic tools has shown great potential in accelerating the breeding process and identifying key genes and regulatory networks that control important traits in rubber trees. So using a data set of about 30,000 SNPs distributed across 408 genotypes, we performed a GWAS analysis in rubber tree. By associating these markers with stem circumference phenotype, we could establish 4 SNPs that were potentially associated with rubber tree growth. However, as an additional resource, we also have modeled a global gene co-expression network using transcript quantification estimates obtained through RNA sequencing. So for those who may not be familiar with the concept of a gene co-expression network, this structure provides a mathematical representation of gene interactions. So the first step in constructing such a network is based on establishing a similarity measure to compare pairs of transcript quantifications. For example, we can use the Pearson correlation coefficient for that. These measurements are then transformed into a graph, and this graph possesses defined properties based on complex network theory. So with this structure, we can represent genes as nodes and their interactions as connections. And through this structure, we can identify groups of genes that are more cohesive. And these genes inside the group may represent a set of molecules that interact and potentially share similar biological functions. So in this context, we have integrated the GWAS findings with the gene co-expression network modeled. By selecting the group of genes containing the genes located in close proximity to the SNPs identified through GWAS, we were able to obtain a more comprehensive set of genes that are potentially associated with the phenotype under investigation. As we were expecting, these additional genes inside this co-expression model were also associated with other SNPs. And interestingly, these additional SNPs found through the association of GWAS gene co-expressions in the gene co-expression network modeled were found to be located in close proximity to QTLs that have been previously described in the literature, showing the potential of coupling these two omics layers together. So the confluence of these findings has facilitated a more comprehensive exploration of the underlying mechanisms that drive rubber tree growth, shedding light on critical biological process. As you can see from this figure, the group of genes studied is primarily associated with the regulation of growth biological processes, as indicated by this main gene ontology terms. However, it is important to note that there may be other underlying mechanisms that contributed to the growth of rubber trees. So in addition to this kind of association, our research group has also focused on the development of genomic prediction models aimed at facilitating the application of genomic selection and breeding programs. In comparison to conventional phenotypic selection, the incorporation of genotype data enables the creation of more resilient models, providing a better estimation of breeding values. And by using a diverse population consisting of multiple individuals, we can create a predictive model to evaluate novel crosses with a high potential to provide more genetic gain in less time. So for rubber tree genomic prediction, our latest contribution was incorporating neural networks in such a task. So using a set of about 400 individuals divided into 3 subfamilies obtained from a real Brazilian breeding program, a previous study from our group has evaluated the feasibility of genomic selection in rubber tree, contrasting different genomic prediction models in 2 cross-validation scenarios. The first one, typically referred as CV1, simulates the prediction scenario of genotypes that were not evaluated in any environment. The second one tries to simulate the situation where certain genotypes were not evaluated in one of the 2 environments used. Clearly, it is more challenging to predict the performance of untested lines in CV1. And as previously mentioned in the published paper, the incorporation of genomic selection in rubber tree breeding would only be viable under the second scenario. It was observed that in the first cross-validation scenario, irrespective of the model employed, the association occurrences were significantly low. In this sense, our first attempt to increase the occurrences of such results, we tested different machine learning algorithms for the predictive task. However, the outcomes observed did not reveal any significant differences. As previously reported across the literature, the conventional genomic prediction methods produced highly comparable outcomes. So as our last attempt to develop an effective model, we decided to take the clear population structure present in our dataset into consideration. So with these 3 populations combined, we created a novel strategy for genomic prediction, which we named Divide and Conquer, drawing inspiration from the fundamental principles of computer science. So this method was based in the following. As our attempts to capture additive effects using all 3 populations together did not produce positive results, we explored an alternative approach, taking into account the genetic architecture of each population. So we developed a single neural network structure consisting of distinct prediction components. So rather than predicting an individual's breeding value solely based on their SNPs, we first predict which population an individual belongs to, and then we use a specific neural network trained specifically for that population to predict the individual breeding value. So in addition to superior accuracy results, our innovative approach enabled us to gain a deeper understanding of the complex genetic interactions underlying these populations. As you can see in this figure, our proposed approach has presented superior performance compared to the previously tested methods. And it is clear that our approach surpasses the others in accurately predicting breeding values. In addition to our work with rubber trees, our research has also focused on the development of genomic prediction models for species with more complex genomes, such as polyploid grasses. Given the importance of sugarcane and forage grasses in our lab's research, we have designed a genomic prediction method tailored to such species, and we named this approach as joint learning. So, as you may know, sugarcane holds a large economic importance due to its diverse applications, such as the production of sugar and also biofuels. As one of the most important crops in the world, sugarcane plays a very important role in the global economy. However, the inclusion and incorporation of genomic tools in sugarcane breeding is hindered by its high genomic complexity. Current sugarcane cultivars were generated by crosses with two main groups of ancestral species, Saccharum spontaneum and Saccharum officinarum, which are themselves genetically diverse. Therefore, the current genetic configuration of sugarcane varieties include chromosomes from officinarum and chromosomes from spontaneum, also including recombinants. And all of this forms a highly polyploid and aneuploid genome. In addition to this, the lack of high-quality genomic resources for sugarcane complicates the identification of genetic markers and poses challenges for the development of genomic prediction models. In the same way as sugarcane, forage grasses also hold large economic importance due to their extensive use in pastures as a source of food for livestock. In addition to their use in agriculture, they have also been explored as a potential source for biofuel production. The high productivity of forage grasses makes them an attractive option for biofuel production, as it could potentially meet the growing demand for renewable energy sources. However, it is important to note that a majority of the species used in Brazilian pastures are polyploid, resulting in a lack of genomic resources. As a result, there is a need to develop effective genomic prediction models to assist the selection of desirable traits and accelerate the breeding of improved cultivar. So considering, for example, a small genomic region with 4 SNPs across 3 diploid individuals. In each SNP, every individual will have 2 alleles, constituting its genotype. In a simple genomic prediction model, we are interested in estimating the mutation effects into the phenotypes. So assuming that for each SNP will have an additive effect E, we can easily calculate the breeding values of the individuals. However, when we have limited information regarding the genomic reference employed, the quantity and quality of the markers will be restricted as well. In addition, when dealing with polyploid species, we'll have additional steps to estimate the correct allele dosages. Furthermore, in the case of sugarcane, we have another layer of complexity caused by the variable ploidy across chromosomes. It's aneuploidy. And we also have diverse positions with a significant amount of missing data. And to compound this challenge, our experiments often involve a target sequencing strategy, which means that we are working with a limited amount of genomic information. For such reasons, researchers have been observing in the literature that the accuracy of genomic prediction in polyploid grasses is generally low. And to address this issue, we created a method that combines classification and regression techniques in order to supply an efficient method for estimating breeding values. So our approach involves 6 distinct steps, which are the trait discretization, feature selection with classification techniques, feature selection with regression techniques, combination of this subset of SNPs identified through feature selection, and linear regression. So the first step, which is the discretization, it involves creating phenotypic intervals from a continuous trait. So in addition to the quantitative trait, we also use the information obtained from the phenotypic intervals for performing prediction. So With the intervals in the quantitative traits, we perform 2 rounds of feature selection to create our model. By using diverse machine learning approaches, we define a subset of SNP markers that might be associated with the quantitative traits and with the phenotypic intervals created as well. Such SNP set is then used for model creation and also for model evaluation. So we tested our approach with 3 different populations of grasses and several phenotypic measurements, and we could improve the predictive capability of genomic prediction in all the scenarios tested. For sugarcane and 2 species of forage grasses, the proposed approach outperformed a conventional Bayesian method, as you can clearly see in this figure, where we compare the real phenotypic values with the predicted ones. So, in addition to genomic selection and plant breeding, the choice of plants for crosses is also an important step that must be performed according to specific breeding interests. Fortunately, recent advances in genomic technologies have revolutionized this process, allowing for the target selection of beneficial alleles that can enhance desired traits. Indeed, it is the synergy between plant breeding and biotechnology that holds the key to unlocking the full potential of future crops. And in this sense, we have also investigated if our developed approach for performing feature selection could provide insights into the definition of genomic associations with phenotypes of interest. So first, we employed the method for investigating sugarcane brown rust resistance. So brown rust in sugarcane is caused by this fungus, and it affects the plant foliage and its photosynthetic capacity. This disease has already caused large economic losses and genetic architecture related to the configuration of resistance is not fully understood. So based on the selection of a subset of SNPs using the feature selection method, from about 14,000 markers, we were able to create efficient machine learning models to predict the plant resistance status. When we compare the occurrences of different algorithms using the entire data set and the selected subset, the increases in occurrences were notorious. And most interestingly, the selected markers were located in close proximity to genes that have been previously linked to brown rust resistance, including the major locus that has been identified as conferring resistance, the BRU1. These results not only confirm the important role of these genes in resistance, but also provide valuable insights into potential molecular mechanisms underlying this trait. Later on, we evaluated the same approach for sugarcane yellow leaf virus resistance. This disease in sugarcane causes leaf yellowing, reducing sugarcane productivity even in the absence of symptoms. So this disease impacts key physiological processes such as photosynthesis, sugar metabolism, and transport. And currently, there are no effective treatments available for this disease, which makes the development of resistant cultivars a critical strategy in sugarcane breeding programs. By applying the same feature selection methodology developed, we have replicated our findings with consistency. Out of about 70,000 markers, we have been able to identify around 200 that present strong predictive power and are closely linked to the resistance mechanism. Finally, we have tested our approach with sugarcane mosaic virus resistance, similar to yellow leaf virus resistance, to the yellow leaf virus. This disease is caused by several viruses affecting sugarcane productivity, and the same observations could be performed. The developed technique enabled the establishment of a very restricted set of markers with potential association to the resistance. And for this last study, we also evaluated the genes nearby the SNPs within a gene co-expression network. By selecting the network modules containing such genes, we could find enrichment for relevant biological processes conferring resistance. And when it comes to breeding stages, it is clear that for certain crops that we are currently working on, we are still at the breeding 3.0 stage, as our primary emphasis lies in molecular breeding. We aim to identify specific genes or genomic regions that are linked with desirable traits. However, we continue to progress towards the era of breeding 4.0, combining our recent advances with cutting-edge data analysis techniques to accelerate the development of enhanced plant varieties. So I would like to thank you all for the attention. Thank you again for the organizing committee, the institutions that are part of these projects, and the funding agencies. Thank you.
Thank you, Alexandre. Thank you so much. Questions in the chat, please, for people that there's already a question that we have here from Enzo. He says, great presentation and interesting work that you have done. My question is, what were some challenges that you found when implementing neural networks to categorize your subpopulations in the rubber tree breeding program?
Sorry, Carlos.
Yeah, you can look at the chat also. The question is there. Okay.
Let me just check. Perfect. So thank you, Enzo, for your question. When I implemented this single neural network structure, the task of predicting which subpopulation an individual was very simple, because as you could see from the principal component analysis, there is a clear separation between these populations. So this task had almost 100% of accuracy. It was very simple to predict it. And when we can predict which subpopulation an individual is, then we can create a specific model to estimate the breeding value that the individual has. And we use only a specific network for the subpopulation then.
You see the next question there from Freddie. What do you think could be the success of using genomic selection in sugarcane, taking into account that the genetics of the species is complex in terms of polygenic characters?
Yes. When I started to work with sugarcane, it was very challenging because sugarcane indeed has a very complex genome. It has the incidence of many duplicated regions, and the traditional quantitative genetics models do not work with sugarcane. So the most challenging aspect in implementing genomic selection in sugarcane is making an effective genomic prediction model. So the key of the models that we have developed is, if we cannot estimate the correct effect of all the markers, we select a subset that we can do it. And this subset was enough to create accurate models. So at the moment, this is what we know. And what we are doing is expanding our tests into more diverse populations to check in which point of sugarcane breeding program is more beneficial to include such strategies.
So I want to follow up on that question. So does the Universidad de Campina have a sugarcane breeding program that you validate all these models and they apply it, and then you can see I would say in real life what is the efficiency of these models? Is that correct?
Carlos, we use data from the Agronomic Institute of Campinas.
Oh, yes.
So we did not test the inclusion of these models in the genomic selection itself. We only used cross-validation strategies and try to mimic the breeding scenarios. So what we want to do now is to simulate how the breeding program is, and then to test whether we can or not include this kind of prediction.
Interesting. Maybe we can get you to another seminar in a couple of years or so and see how it went. Question, Chao is interested in it says interesting approach to use this technique to find target genes for gene editing, for example. Have you collaborated with anybody in that context?
Yes. We are now working on selecting target genes for CRISPR. So I cannot supply many details, but our strategy has the potential to do it as well.
Yeah. You have a question from Isaac here. Great presentation. Are there plans to develop sugarcane cultivars that are resistant to the viruses, knowing fully well that we don't currently have a treatment for it? Are there current works on checking the extended impact of vectors that have not impact of vector have on the spread of the virus?
Yes. So as I presented to you, this work with viruses is from a colleague of mine, Ricardo Pimenta. So I do not have much knowledge about viruses, but there is a growing interest from the Agronomic Institute of Campinas to develop this kind of cultivars, because indeed there is no treatment for that. So if the disease comes to the field, then there's nothing they can do.
Okay. In your question from Roshan, in your experience, what was the sensitivity of your machine learning models for the number of individuals within a given subpopulation?
Yes, this is a very good point. What we have observed is when the number of individuals in a population is small, the models are better. If we start to increase the number of individuals, the models start to decrease in terms of accuracy. So what we did to test the feasibility of our approach for larger populations was simulating a bigger number of individuals and testing if there is a maximum number of individuals that the model can work. And what we see is that if you use, for example, 1 million of individuals, our approach will work exactly as the traditional genomic prediction methodologies. But if you have this large amount of individuals, the traditional methodologies will work. They will have enough accuracy to predict. So our problem was to make the models work when we have a restricted number of individuals. So yes, this is a very good point.
We have a question from Simon. Thanks for the presentation. Can the genomic prediction model you develop is it specific to sugarcane, or it can be extrapolated to other commodity crops?
Yes, it can be. It can be. We already tested the genomic prediction method for pine. We have tested for wheat, and for maize as well.
Mhm.
And all the crops that we tested, the results were very positive.
I want to ask you a follow up on this one. To use can you use comparative genomics from, I mean, from networks of genes that, let's say in corn, that have been heavily studied? Can they help you in your prediction if you use that publicly available information with somehow related species?
It can help, Carlos, if you include into the network an additional layer, which would be, for example, a layer of transcriptomics data or proteomics data. And this is something that we are working on as well. How to include intermediate omics layers into the prediction models. It would be like a multi-omics model to predict phenotypes. And then we could, for example, use this information from other species to create our network. And it would be beneficial. Yes, it would be really, really nice.
Okay. He was asking, what do we mean by selecting target genes for CRISPRs? Can you elaborate a bit more how you can go to the gene itself and discover it and then target it?
Mhm. As I told you, we are interested in selecting SNPs that are associated with a phenotype, right? So if we know that a genomic region is associated to a specific phenotype, and we know the genes that are within this region and how they respond to, for example, a disease, we can supply a target to be silenced by CRISPR, for example, right? So instead of only evaluating the gene expression levels, you can couple this information with genomic information as well and have a potential target for being modified. This is what I meant by selecting targets.
Any other questions before we close the seminar? I wanna share the screen with hold on a second. Now with the right title for the seminar. Okay. Apologize, Alexandre, for that problem. I did not check correctly the first slide. So thank you so much, Alexandre. This is the first time, I would say, that we have advanced PhD graduate students presenting in this seminar series. And I think you have done an excellent job, Alexandre. And it sort of has led us to discuss the possibility of creating a whole series, or at least half of the series, with advanced PhD students or postdocs presenting in the future. So you guys will hear more from me. It requires quite a bit of coordination, but I think it's excellent to have this platform, this forum, to expose the work that you guys are doing as Alexandre is doing. Thank you so much for what you have brought to us, Alexandre. Next week, Thursday, is the 5th seminar. So we have 2 more to go by Dr. Ruben Alvarez Reyan. He works here at NC State, and he is going to be talking about lessons from the genetic of local adaptation of native maize varieties and wild maize relatives. So please join us at the same time as today, next Thursday. Again, the recordings will be at the bottom of the announcement that you guys will receive tomorrow, reminding you of next week's seminar. So if you scroll down to the bottom of that announcement, you will have the link to the recording, okay? Thank you so much to Thiago and Brandon. Brandon, unfortunately, he's sick and he was not able to join us, but he's always very helpful in organizing and running the seminar series. Thank you so much. You guys have a great weekend, and I look forward to seeing you again next week. Thank you, Alexandre. Muito obrigado. Tchau.